It's just not a matter of, I do biotechnology, so you might say, oh, well, we can do genetically modified organisms, isn't that a cool technology? But in some contexts, that isn't going to work. And so being able to apply that social lens to it also allows us to come up with better solutions. So I have a variety of different projects ongoing, and the projects differed based on what the students are interested in. One that I'm really excited about right now is one in which we're working with some high school students in the local schools. It first originated with an ISAT senior who was working on his senior capstone project, and he was really interested in agriculture in general and the use of nitrogen fertilizers. So we started making the collection in about 2013. And in that process, we realized that it was just going to be too slow. If we were only collecting 4 or 5 strains a year, it was going to take us forever to get a collection big enough that we could actually make any conclusions about it. At the end of that project, we got the idea, well, why don't we partner with some high schoolers who are interested in doing genuine research? And then this year, I've got 2 students working with me, and we are really just developing that even more.
So we started this fall by visiting a local high school classroom, and so we went with them on a field trip out to local farms. And so there we showed them how to collect soil samples, which they then used to inoculate soybeans, and they grew those up and are now ready to extract the bacteria from inside the nodules.
And we did this as part of an outreach with the local high schools as well. We wanted to get involved with the local science community. You think of a stereotypical high school classroom and they check out, but these kids were really interested in what was going on. They seem invested in the whole process. We made a video tutorial for them showing them how to inoculate their soybean seeds with the bacteria that we're interested in. We showed them how to make these mini incubators for the soybean plants called liner jars, and we will be working with them in harvesting the nodules that grow on the roots of the soybean plants where the bacteria lives.
From there, we can isolate the bacteria, and then the ISAT students work on the genotyping, so looking at what genes are found in those bacteria that we collect. The high school students get the benefit of being able to be plugging into a real research program that means something, so this is a year-long effort. And so it really is a true partnership because the high school is getting things and we are getting things, and it's our own little symbiosis. Moving forward, I would be guessing that we would have some students that would be focusing on the genotyping that happens in the lab, other students would be focusing on the outreach, and others that would be transitioning between those venues.
Within one group, you'll have different concentrations and different majors, and so you'll have to learn how to communicate between each other, coming with a different background of knowledge. And so what usually happens is you have a more robust and better project or end goal to present because you have so many different perspectives on the assignment.
That's the emphasis. And then you say, well, what tools do we have to address that problem? And that's where the disciplines come in. And I think that's just inherently a different way of thinking about the way that science is run. You don't just do biology because biology is interesting to you. You do biology because it can solve human problems. So if you have that sort of perspective in that you want to do good in the world and you want to start with that problem and see how you can fix it and then use the tools that make sense to do it, ISAT is the place to be.